So for my research today, the title is Isolation of Local Soil Cultures to Screen for Antibiotic Producers. This was done by me, Abigail Cunningham, along with my faculty, Dr. Volkanis. So to get started, antibiotic resistance is one of the most prevalent and terrifying changes in medicine that is in our current world. Researchers and scientists from all over the globe have dedicated their lives to studying antibiotics in general, the creation of antibiotics, and the resistance that bacteria can build up to common medications that are used to cure or lessen the symptoms of. Based on the State of the World's Antibiotic Report conducted in 2015, over the years, some countries have leveled off in the amount of antibiotic resistant bacteria, but numbers still continue to rise in countries that are considered lower and middle income. With the ongoing issue of antibiotic resistance, it is more important now more than ever that we utilize our resources in the world around us. Antibiotics come from many different things, such as fungi and soil bacteria, and many, I feel like, do not realize the benefits that these microscopic organisms may be able to present us with. By testing local soils from all over, we can find out whether the bacteria growing in the soil right in our own backyards could have the potential to be transformed into a life-saving antibiotic. Once we test them and input our results into an online database, or we share results online, we are able to create a pathway of scientific discovery that could lead to scientists and researchers from all over the world wanting to test local soils cultivated and tested by students that could possibly help the greater good. It is hypothesized that through the testing of our local soil, antibiotic producers will be isolated and therefore become successfully identified. Our materials and methods. So throughout this entire process, we used the Tiny Earth Lab Manual, and we used multiple different techniques sort of throughout our weeks to complete this. But by using the Tiny Earth Manual, we were able to carry out each experiment as accurately as possible. First was serial dilutions. That was the first experiment we performed where we used 1 gram of soil, transferring it into a test tube, adding 9 mils of water, and then performing subsequent dilutions in order to obtain a more dilute sample for our spread plates. Once completed, we resorted back to the Tiny Earth Manual, where we utilized proper spread plate procedure. After our results were obtained from the spread plates, we then went back to our Tiny Earth Manual and we used it in order to complete our streak plate. And this we did in order to obtain isolated colonies through diluting the inoculated bacteria on a single plate. Then we looked at all of our morphology, and we wrote down our colony morphology, and this was used to study the bacteria in order to choose bacteria of different color, texture, shape, and that way we would use all of these for our master plates. We used all of our serial dilation plates, which was 6 plates, but down the middle. So there was technically 12 different things that we were looking at, and we chose bacteria, like I said, based on their texture, their shape, their color, just that way when we created our master plate, they were not all of the same bacteria if we kind of had anything to do with it. That way, you know, we were able to pick and patch a more diverse selection of colonies. So then our results were recorded the following week, and then we used it to pick and patch colonies based on morphology for our master plates. The picking and patching colonies protocol was also from our Tiny Earth Manual, and it allowed us to carefully examine organisms on the streak plates in order to choose unique organisms to be grown on the master plate. After we inoculated our isolates from the streak plates, the bacteria was then put through a gram stain test in order to compare unknown bacteria from soil isolates to known bacteria and their morphologies. That way, you know, when we were looking at known bacterias and observing their shape and whether they were gram negative or gram positive, we sort of had a basis for what we were looking for when we were observing our unknown bacteria through the course of a gram stain. Once results were obtained, the plates were then screened for isolated antibiotic producers. Bacteria was then inoculated from the streak plates to test for the presence of lactose and non-lactose fermenting bacteria through a series of agar plate tests. The two types of agar that we used and their protocols, EMB agar and McConkey agar, those are also in our Tiny Earth lab manual. We pulled everything directly from it. That way we could follow it as accurately as possible. Lastly, the final tests that we did we used the isolated soil samples and put them through a variety of biochemical tests. Again, we followed protocols that were listed in our Tiny Earth lab manual, and these steps were followed to perform the Oxidase test, Catalase test, and a gelatin test on the isolates from spread plates in comparison to known bacteria. Our results. So for the soil collected, this was entirely collected outside of my apartment in Hopwood, Pennsylvania. The top layer was scraped off and soil with a clay-like texture was collected approximately 1 to 2 inches deep. The soil was then used for a series of subsequent serial dilutions for spread plates. When observing the spread plates, growth was witnessed on all 3 of our plates. The most prevalent, abundant growth was on the 10 to the 3rd plate, since it was the least diluted. For when we looked at these results, we looked at our 10 to the 3rd, our 10 to the 4th, and our 10 to the 5th dilution plates, and they became more diluted as you went through. So our 10 to the 3rd was the least diluted, and our 10 to the 5th was the most diluted. So like I said, our most prevalent abundant growth was on our 10 to the 3rd plate, with sort of a moderate amount found on our 10 to 4th plate, and then our 10 to the 5th plate had exhibited the least amount of growth, but the conclusion that could be drawn from that was because it was the most diluted plate of all. Bacteria reported and shown on all of these 3 spread plates were different shapes, sizes, and colors all over. Colony forming units were then calculated based on our spread plate results, and they are as follows right here in my results section. The diluted bacterial samples from the spread plates were then used in order to complete 6 street plates with 12 total sections of bacteria, since the agar plates were split into 2 sections. As I said before, colony morphology was observed and recorded from the 12 different sections in order to identify any differences in the different single colony formation. That way, you know, we sort of had a diverse group that we were moving onto our master plate. From these plates, like I said, the master plate was created. The master plate was to be cultivated from a variety of unique bacteria of different shapes, colors, sizes, so that way we could test a multitude of different bacteria for the possibility of antibiotic producers. Next, we used our unknowns as well as E. coli, Staph epidermidis, and E. aerogenes to test different capabilities of antibiotic producer screening plates, all of which who had no antibiotic producers. Over in the top right corner of my results section, the top picture on the left side, up and down, where it shows things are sectioned off, that was my antibiotic producer plate for the unknown organism. And as you can see, there was no antibiotic producers on the plate. An antibiotic producer would result in a halo growth formation, not a growth formation, but sort of a halo ring that is around the bacteria. That's what you would look for when you're looking for antibiotic producers, but as you can see, there is not one in that picture. Our unknown bacteria was then tested, as well as 2 known bacteria, E. coli and Bacillus subtilis, and their ability to ferment lactose on EMB agar. The unknown and B. subtilis were non-lactose fermenting, and E. coli was lactose fermenting. As you can see right here on the left side, right below the plate that I had just spoke about, is a plate with sort of a reddish agar, and our unknown bacteria was not lactose fermenting. There is no pink smear. You cannot see anything. Therefore, our unknown organism was not lactose fermenting. Next, our unknown as well as multiple known bacteria were put through a series of biochemical tests. First was the Catalase Test, which tested the bacteria and their ability to produce gelatinases, which liquefies gelatin. That would be the picture on the top right, where I have the tube in my hand. In order to test for if it liquefied gelatin or not, we would take the tube and we would tilt it to the side. The unknown organism did not liquefy gelatin, or it did not liquefy the gelatin. The gelatin was still thick and it did not move, therefore it does not produce gelatinase. The next step was the Oxidase Test to identify whether the unknown and 2 known bacteria possess cytochrome C. A positive result for a cytochrome C test would result in a dark purple color. And for this one, none of our 3 bacteria were positive for cytochrome C. This is the top photo on the left, where it is the piece of filter paper that is split into 3 sections. None of our 3 bacteria were, none of them tested positive for cytochrome C. Lastly, we performed a catalase test on our unknown isolate and the 2 known bacteria, which would result in a bubble formation when hydrogen peroxide was applied. And that is the bottom left picture, right underneath the agar that has the pinkish reddish color. And all 3 of those organisms produce catalase activity, including the unknown. The picture over on the far left above my introduction, that is another one of the, that is my master plate for my unknown organism, where you can kind of see that there was no antibiotic producers. So in conclusion, the purpose of this study was to test local soil samples for antibiotic producers. And to do this, we performed a multitude of experiments that would enable us to dilute our samples into single colonies in order to test the most purified samples. Antibiotic producers are organisms that have the capability to be used for the production of different medications. Soil is a breeding ground for microorganisms that can produce antibiotics, and that's why it is more important now than ever that we test these microorganisms located readily available to us. We can do this constantly. We can do this by constantly using our local resources to test different areas of soil for producers, because if positive producers would be shown, it could change the world of antibiotics and potentially present us with something that could really help the world of medication. My results did not show any signs of antibiotic producers, but that could be for a multitude of reasons, such as there were no true antibiotic producing organisms in the soil, or it could have been the result of human error. As for my hypothesis, the results did not conclude what was first believed, but this is why aseptic technique must be followed to the greatest extent to ensure human error is minimal.